Hello everybody, and thank you for your interest. I'm Emmanuel from the Transfusion Center in Epernage in Switzerland, and I work on red cell concentrates. Regarding the transfusion of red cell concentrates, a debate has raised if the donor sex may have an impact on the transfusion patient's outcome, but observational studies could not provide a clear answer. So here in our study, we aimed as a first approach to evaluate the impact of the donor's and the recipient sex on red blood cell properties. And for this purpose, we developed an in vitro transfusion model that you can see here. On the one hand, we used red cell concentrates, two female and two male ones, and we extracted red blood cells by centrifugation and washing, and then we mixed the two red blood cell pellets of the same sex together. On the other hand, we had a pool of female and a pool of male plasma, and these pools represent the recipient. So we incubated red blood cells in plasma in a sex-matched and in a sex-mismatched manner at an hematocrit of only 5%. The idea here was to have a high volume of plasma, to have a sufficient quantity of glucose, and to limit the effect of waste accumulation, because here it was a closed system. Then we incubated the reconstituted blood at standard conditions. Then we sampled at different time points from 0 to 48 hours of incubation, and we split the samples in red blood cells and plasma. And so we started this experiment with red cell concentrates at storage day 2, and we followed these specs until storage day 42. Here in table 1, we listed some standard blood parameters just to validate the quality of the used red cell concentrates and to indicate that red blood cells were maintained during in vitro transfusion. Here in figure 3, we continue with in vitro transfusion. So we incubated red blood cells at storage day 2 and at storage day 42 from 0 to 48 hours. And here the color of the lines show the sex of the red blood cells, and the continuous lines represent the sex match, and the dashed lines represent the sex mismatch. And we can see that glucose was maintained. And here the difference was just due to processing, since women have a lower hematocrit, so a higher volume of plasma was mixed with the glucose containing anticoagulant solution. And here we see that lactate correlates with glucose. Concerning the 2,3-DPG data, we can see a clear rejuvenation process in contrast to red blood cells stored in red cell concentrates. And this rejuvenation process was lower at storage day 42. Consequently, old transfused red blood cells may be less efficient in vivo. ATP was also maintained during incubation at higher levels than in stored red blood cells. And here the sex difference may be due to physiological sex differences, may be related to a different oxygen saturation, but this difference is offset due to same storage conditions. And interestingly, this difference is maintained during incubation here. Finally, we can see that hemolysis increased during incubation and also with storage time, and hemolysis was higher in male red blood cell population than in female red blood cell population. And so in conclusion, this in vitro transfusion model works well, since red blood cells were maintained in an active state at 37 degrees during 48 hours of incubation. And here we notice sex differences in ATP levels and also concerning hemolysis rate. So thank you for watching, and goodbye.